her committee. So Rebekah got her undergraduate degree, her bachelor's in chemistry, with also minors in math and philosophy from Lipscomb University in 2020. And then she made her way here to the University of Kentucky. One thing about when Rebekah came to the University of Kentucky, she essentially said, "I'm coming here and I'm working for you." So it wasn't so much like, hey, let's go through the interview process and all of this stuff. And I was just like, and this was the summer before, even like she and Jared, wherever Jared is, came to visit. Because Jared at the same time was looking for to pursue med school, and they were making those decisions. And so I was told that this was going to be my existence for the next 5 years. Which I think for some people in the room understand this. To say that Rebekah has done a lot would really be an understatement. And this really spans a lot of life of being a graduate student. So she was a TA. She started doing research the summer before coming in. She was a TA. She served as president of the Chemistry Graduate Student Association, where she led a lot of really interesting initiatives for the graduate students. And then you get to the research, right? And so far she has 8 papers published, and I think 6 as first author. There are 3 others that are submitted. And I think you miscounted. I think there's at least 5 that are in preparation. So there's going to be a lot of work that comes from this PhD. And what's really interesting, I think, about that is that, a lot of times as PhD candidates, right, we study a topic, right, and we publish on a topic. But what you're going to see today is that Rebekah has a very diverse span of her science. And so it's going to include some philosophy of science, right? It's going to include electronic structure theory calculations. It's going to include data construction and machine learning and automation and robotics. Okay, so it's really touching on a lot of areas where science is moving, right? And the totality of her dissertation, I think, is going to be a really sort of, you can look back at it in a few years and see it's going to be a really pivotal, right, in terms of how we think about chemistry and materials chemistry in particular. For all of this, and I should also say she also has a paper in chemical education, just for fun. But for all of this, she's received a lot of sort of best poster presentation awards, travel awards to conferences. She's a Lyman T. Johnson Fellow. She got the Steckler Award for outstanding research as a graduate student, and she's also received a fellowship from PEO International, where she's a PEO scholar for this last year of her PhD. So they're not here to listen to me. They're here to listen to you, Rebekah. So I'm going to turn the floor over to you. But thank you for the last 5 years, and I think we're all going to be excited to hear what you have to say.
Thank you so much. All right, can everyone hear me? Can everyone hear me in the back? Yeah, thumbs up. Okay, awesome. So let's get started with the chemistry. So today I'm going to be talking about my work that mostly involves electroactive pi conjugated systems. Let's see if my clicker will work. Maybe.
You need this one.
Maybe.
She's only had it for 6
There we go. I have. All right. So electroactive pi conjugated molecules are essentially organic systems that have pi conjugation, meaning they have alternating single and double bonds, which allow electrons to disperse throughout the pi orbitals of these systems and give them some very unique electronic properties. And I'm specifically going to be discussing electroactive systems, which in general just means a system that undergoes some sort of change when exposed to an electric field. Most of the stuff I'm going to be talking about is specifically redox active, meaning it undergoes a redox event or the addition or removal of an electron when it's exposed to an electric field. So these are some just very basic examples of the type of systems, and they have a lot of applications. Anything from semiconductors to biosensors, photovoltaics for energy generation, energy storage applications, all over the map. And so these are really, really interesting systems in materials research that have a lot of promise. So why is this? Why are these systems good for these applications? Well, first, they are organic. So generally that means they're less corrosive and toxic than their metal alternatives. In a lot of cases, maybe not all, right? And they can often be synthesized from more abundant and cheaper materials, which lends itself to greener chemistry. And then the really big thing that is good about these systems is they have very tunable electronic properties. So what does this mean? Well, if we take an example of an energy storage application, flow batteries, which is something I'm going to discuss a couple times today, in these applications, you want a molecule that is highly soluble, that has a high oxidation potential, that has multiple electron transfer events, and so on. And if you start with a class of pi conjugated molecules like this phenothiazine, this is a group of molecules that was synthesized in large part by the Odom lab here at UK. You can make small structural changes to the molecule and optimize a specific property. So these methoxy groups can allow for 2 electron transfer events. You could make another change and increase the solubility. It's also possible to optimize multiple properties at the same time. So you can do both solubility and the number of electron transfer events, or the number of electron transfer events and high potential, or even if you're a very good chemist, you can try to optimize all of the properties at once. So you can see how this lends itself to being able to design a molecule for the exact application that you're interested in, right? But at the same time, you can see that small structural changes have significant impacts on the properties of the molecule, which means there are so many possibilities for the types of systems you could create. And that leads to a massive chemical design space. Now, of course, informed chemistry and chemical intuition can help you navigate that. But the space is so large that it can cause some problems. So for context, there are 10 to the 18th grains of sand on the Earth. There are 10 to the 23rd stars in the visible sky. And there's estimated to be 10 to the 60th possible small organic molecules. Now, this paper where they computed these figures were specifically for pharmacologically active molecules. So the real search space for materials is probably even much larger. And you can see this diagram here kind of shows a visualization of chemical space and compressed dimensions. And there's a lot of areas where we have a lot of information, a lot of published literature, but there's a lot of white space in between. There's so many areas of chemistry we just haven't even touched, and many areas that are sparsely populated. So there's a lot of potential for new things. And I'm going to argue throughout this talk that this massive design space requires data-driven tools and automation for the discovery of new molecules in an efficient manner. So it's no secret that AI and data are transforming the world, science, and chemistry specifically. For example, the Materials Project, many of you may be familiar with, is a database of materials, both organic and inorganic, that's been called the Google for materials research. It's led to many different innovations. The AlphaFold protein structure model has been able to very well predict protein structures from a sequence of amino acids. And increasingly, we're seeing large language models like ChatGPT used in advancing chemistry, from reading the literature, generating synthesis pathways, and so on. So we're really at the cusp of new chemistries and new ways to apply all these applications. And these different tools can be used to help better explore chemical space. So automation is a key component of this. If we imagine maybe three-dimensional representation of chemical space, there are many different ways that we can use automation with these data-driven tools to help better explore. We first need to be able to generate a chemical space, some sort of virtual representation of where things are and how close different molecules are to each other. So you need automation for that. Predicting different properties for the molecules, validating testing parameters for when we design property prediction models and generating chemical space, and rapidly exploring this space are all important ways that we can use automation to help better explore and find new molecules. But the key thing is all of these different applications are founded on good chemical data. So to train any sort of data-driven model, you need data that is well structured and well curated to produce accurate results. There are many different ways that you can generate chemical data. Maybe the most obvious one would be traditional experimentation, where you create a molecule in the lab and then you characterize it in some way to find properties of interest. We can also use quantum or classical physics to simulate what a molecule will do in virtual space and estimate some of the properties of interest. And then if we already have some chemical data, we can train machine learning to very quickly estimate properties of interest for any given molecule. And you'll see here that the timescales vary pretty significantly between the different methods. And you might think, oh, we should use machine learning all the time because it's the fastest. But at the same time, experiments often better reflect the observations we're going to see in the real world because they are closer to the material applications where these molecules will be used. So while you can get really fast results here, these may better reflect real world observations. So there's a balance. There's a place for all of these different methods of chemical data generation in science moving forward. So today I'm going to explore big data generation, analysis, and the infrastructure that's going to be needed to enable automation and data-driven discovery for these pi-conjugated electroactive systems. And I'm going to hypothesize that efficient discovery of these systems will require a coalition of both automation and data-driven design, coupled with chemists themselves and chemical intuition. And that also necessitates large-scale query data and human-machine interfaces. So I've begun with a bit of an introduction, some background on the problem space, and then I'll jump into a discussion of how we might organize chemical data. After that, I'll give a real-world example of how we can build and populate a chemical database. Then a few examples of applying such data before I'll talk about how we can generate large-scale experimental data with robotic experimentations. And I'll close with a perspective. Okay, so first, if we're going to be building large-scale chemical data, we need to think about how we're going to organize it. Are we going to organize it in a way that is useful, that is informative, that avoids certain biases? And so we need to think about the database schema. So what is a database schema? Schema is a database term for the structure in which the data is stored. So if we imagine a database or a set of data on crystal structures, we can imagine that each entry in that database would be a single crystal. And maybe you have associated crystal properties for that crystal, and then also a list of molecules that are in that crystal. And each molecule will have a list of associated properties. Similarly, if we have a database that has mostly solutions, we could have a schema where it is a solution-centric schema. So each entry is a solution. You have a set of solution properties, and then maybe a set of molecules that are solvents, a set of molecules that are solutes. Each one has associated properties with it, and so on. Or if we wanted to incorporate all this data together, we might have a molecule-centric database schema where each entry is a molecule, and you have molecule properties and then a list of the crystals and solutions that molecule has been recorded in. And this all encompasses most of the same data. So the question is one of these correct? Is one of them just universally better? Now, the broader term that's used in philosophy and history for this way we organize data is ontology. So the big question is there some universally correct ontology that we should be using for chemical data structure? And if there is, should we use it, right? And there's a little bit of discussion in the field about this right now, maybe not as much as there should be. But for the most part, it's kind of constrained to within chemistry and amongst chemists. So we wanted to take an interdisciplinary approach to answering this question. Should there be a universal chemical ontology? So we collaborated with a philosopher of science here at UK to take a stab at this question. And we began by looking at the historical context of data science and classification and informatics and chemistry. Then we explore some of the philosophical concepts that are important for answering this question with chemistry terminology, so chemists can understand. We then explain some of the philosophical arguments for whether or not we should have a universal ontology, and then argue with many philosophers of science that we should actually take a pluralistic approach to chemical ontologies. And I'll discuss that in a minute here. But finally, we did want to advocate for not only a shift towards more pluralistic ontologies, but also just more collaboration between philosophers and historians and chemists, because they have a lot to say on these big questions that we need to answer if we're going to move forward with data-driven chemistry. All right, so we began with the historical context. And so we looked at how throughout the history of chemistry, chemistry has been a leader in thinking about how to classify things as atoms or as elements or so on. At the same time, chemistry has also been a leader of how we compile and distribute data. So the original handbooks and databases for chemical abstracts and things like that were actually at the forefront of data science and the forefront of chemistry. And chemistry has also been at the forefront of informatics, using that data for some application. And so it should be no surprise that chemistry is now, again, at the forefront of asking questions about organizing large-scale data for machine learning and AI applications, because chemistry has been here before, right? And chemistry is poised to be a leader in the sciences when answering these questions. So we wanted to look at some of the philosophical concepts that were relevant. Chemistry's been asking these questions for maybe a few decades, maybe a couple centuries. Philosophy's been asking these questions for millennia. So they have some frameworks that can help us understand the question and the options for the answers. So the first for that is monism, which is the idea that there's really only one correct scientific theory to describe a system. A monistic answer to the question, is there universal ontology, is yes, there is. We should use it. So an analog in chemistry might be the IUPAC nomenclature system. So most chemists will know that this is the way that the community has decided we should always name systems for the most part, right? And it's a great system for naming molecules. It's descriptive and it's great for human users. And this might be the monistic way to have chemical nomenclature, right? But the opposing chemical or philosophical idea would be pluralism, which is that there may be multiple theories or models that can be used to describe a system, and they're all okay in their correct context. So in one context, you might use one, in one context, they might use the other, and on the whole, they provide a better picture of what's going on. And so when it comes to chemical nomenclature, we also have SMILE strings, which have come up more recently. And they really give the same information as a IUPAC nomenclature, but they're much better for digital users. And they're much more efficient for inputting molecules for data applications. So in one context, SMILE strings is better, when one context nomenclature is better. And there are other examples of pluralism that exist in chemistry. This is just one that may be useful. So skipping the majority of the argument of the paper in which we describe this out, we ultimately argue with many philosophers of science that we should have a pluralistic framework for chemical ontologies. And we have various different arguments to support this claim. One would be that there is some evidence in sociology that having a pluralistic mindset in a group of scientists can help those scientists arrive at truth more quickly. That's supported by some historical evidence that major advances in chemistry in the past, such as the development of the idea of conjugation itself, arose from maybe slightly opposing different theories that were seen together. And also, we talk about some existing problems that exist in database ontologies right now that already kind of attempt to form some sort of universal ontology. And there's just some ways in which they can't capture everything all at once. And we ultimately argue that the current diversity of ontologies that exist in the field is not a problem, as some people say it is, that we should not waste more time and energy trying to create a universal ontology, and instead focus on context-sensitive, function-focused ontologies that are then interoperable with each other, so that we can use data more efficiently moving forward for the design purpose. So now that we have a foundation of how we can organize data, let's discuss building a database in real life for chemical data. And just a little bit of context here. I've been involved in the Details project for most of my PhD, which is an effort to accelerate the discovery of small molecules for liquid-based energy storage, or flow batteries. And we wanted to build a database of potential molecules for this application, electroactive high conjugated systems, that contained computational data that was generated through a high throughput quantum chemical workflow, along with experimental data. We had several collaborators in Iowa that ran experiments for us and recorded data on these systems. And we also wanted to enable some large-scale experimental data with robotic experimentations, and try to read some information from the literature to include that in our database. All of this was designed to build a data structure where we could then perform machine learning to better inform the discovery and the development of these new systems. And we wanted to make sure at the same time to share all of this with the community. So these are the areas that I have been involved with. I constructed the database itself, constructed and monitored the high throughput workflow. I've been involved in the robotic experimentations, some of the machine learning applications, and sharing the information with the community. So the database itself uses a molecule-centric schema because this is a molecule-focused project that contains general molecular information about the molecule as a whole, as the molecule species, as well as some experimental data. We tried to make it a function-focused schema, and then also abide by fair data principles. And then the web interface associated with the database enables users to search for a molecule by ID, by the source name, smile string. And then for each molecule, there's an interactive viewing web page where you can see all of the computed properties on the systems. This right here shows the user base we have for the Details website. So lots of different countries are involved, about 1,000 users currently. And then we coupled all of this with an application programming interface, or an API, so that users could interact with the data through a Python program. So that would make it more easy for the users to utilize the data for data-driven applications like machine learning and such. In populating the database, we began by scraping some potential structures from some big data sources, such as the Cambridge Structural Database and Zinc. We also accepted user submissions and had some molecule generation algorithms based on common motifs in the literature. We then filtered all the collected data by some rough filters. We wanted to make sure the systems had 5 or 6 rings usually, or 5 or 6 atom rings, usually 1 to 4 rings, and less than 3 heteroatoms in a ring. And then in order to actually get to a reasonable number for all the calculations we wanted to do and the computing time that we had, we narrowed down this 115,000 molecules to 43,000 by finding the most diverse group of molecules. So the idea was to create a large and diverse chemical space as opposed to a really compact and dense chemical space. The merits of this can be argued later on, but this is the idea here. And then we computed full redox potentials. So you can see the general distribution of molecular weight for the systems. Most of them reside around 300 to 400 grams per mole. Some of the molecules are bigger. They don't quite fit through this filter. That's because they were user submissions, and we did allow that for users who wanted specific molecules to be in the data set, mostly for these flow battery applications. The redox profiles were computed via a fireworks-powered high-throughput quantum chemical workflow with density functional theory, and this included molecular level descriptors such as HOMOs, LUMOs, redox potential, absorption spectra, geometries, and then also some species level descriptors. And then this right here shows just a start of how we can analyze the data. There's not anything groundbreaking here, but just how we can begin to use this data to infer something about the chemistry that's going on. So we can see just as you have higher numbers of atoms, so bigger systems, you generally have larger monocation radical stability scores, which makes sense. There's a bigger molecules, more space for that ion to delocalize. So this is how we can start to use this data for analysis. But there's lots of other things we can do with the data than just plotting it all on a scatter plot, right? So what can we do? Some of the applications we have begun include traditional analysis like the one I just showed you, machine learning for classification of data, machine learning for predicting actual scalar properties. We can also use large language model type things for building encoders in order to generate a chemical space that we can explore. We can use machine learning algorithms and the properties themselves for funnel type pipelines to narrow down search spaces. We can validate existing measures and metrics and so much more. So we've begun all of these things, but today I'm just going to focus on two of the ways we can use chemical data. So first I'm going to share about how we can use machine learning and chemical data to build classification models. This actually does not use the details data. It uses a similar data set of electroactive pi conjugated molecules that was generated in a very systematic manner. So we had these monomer units. They were all 2-ring monomers that were extended up to 14 rings for short oligomers, and then substituted with various constituents. And the purpose here was to study the central dihedral rotating angle in the middle of the oligomer. So we scanned the potential energy surface of rotating that angle all the way around to see what kind of energy surface we'd get. And the first thing that we noticed when looking at the data from all of these calculations, it was about 270 different potential energy surfaces, was that all of the surfaces kind of fell into 4 main peaks, being a rolling hill with a relatively low-lying energy peak, the central peak right here, an asymmetric peak, or a W valley type or energy surface. And so because all of the surfaces fell into one of these 4 categories, we thought we might be able to do something with this that would make it easier to predict an energy surface based off of just a 2-dimensional structure. And so I built a machine learning model that takes the SMILE string with a 2-dimensional structure for any monomer and will predict the shape of the potential energy surface for the central dihedral angle. And this right here is a confusion matrix that shows that on the testing data, the model did a fairly good job of correctly predicting the category of the potential energy surface. So this is interesting on its own, useful. You could take a molecule without having to run any calculations, predict the surface. But we wanted to know if we could take it further, make it more applicable. And so I found this existing analytical analysis that had been published previously, where they begin with a SMILE string, their own analytical analysis, and the input of a DFT calculated potential energy surface, and use that to estimate some more mesoscale properties of the oligomer systems if you extend the units very far, to find things like persistence length and mean squared end-to-end distance for these longer molecules. So these are more complex properties that are harder to calculate even with full DFT. So this is a useful method. It does require the computation intensive potential energy surface to perform the estimation, but I replaced that with my machine learning prediction, which takes a couple seconds per molecule. And I automated this entire process with a Python package that is now published and available to the community. And ultimately, we were able to very well produce with our machine learning predicted surfaces the DFT predicted calculations for these mesoscale properties, for the persistence length here and the mean squared end-to-end distance. So this is really useful for bigger applications that we can do this prediction relatively quickly. And it is worth noting that the original analysis was experimentally validated. So I have fair confidence that the predictions with the machine learning algorithm will reflect experimentation as well. All right, moving on to the next application of data. We can use big chemical data to validate existing measures and metrics. And the one I'm going to focus on today is molecular similarity. So if we have two molecules, like these two here, they're similar in some way. And think about a broader application. If we were building a chemical space, intuitively, we want these two molecules to show up close to each other because they're similar. So we want a way to test, okay, if these 2 points on chemical space are similar, are they actually similar molecules? And there's several ways we can measure quantitatively whether or not two molecules are similar. But one of the most common is using some sort of fingerprint generator to turn these structures into bit strings, which are easy for a computer to use and analyze with math, and then use some sort of distance function to compare the similarity of those bit strings. Now, what kind of fingerprint generator to use and what type of distance function? It's a wide variety of options here. And so we wondered, okay, is there a best one? And specifically, is there a best one for capturing the relation of electronic structure properties? So these two molecules are similar, and because of that, they have relatively similar HOMO energies. And if there's some similarity in structure that causes a similarity in electronic structure properties, theoretically, that similarity structure should also cause a high similarity score. So theoretically, there should be some relation between the similarity score and the similarity of the electronic structure property. And so to do that, we use the details data along with some other data sets to measure how well 40 different similarity scores, some combinations of fingerprints and distance functions, capture electronic structure theory. We began with about 26,000 molecules with full electronic structure profiles, and then did a pairwise comparison of each molecule for all of the different similarity measures, which gave us about 350 million molecule pairs. And for all of the similarity measures and all the electronic structure properties, that's about 16 billion data points. So this is not a trivial amount of data. Just moving it around caused lots of headaches. And we actually, in order to do the analysis to figure out how well these similarity measures captured electronic structure properties, we had to develop our own metric, kernel density estimation-based metric, because there's some confusing things involved. And we then developed an efficient sampling method so that we didn't have to use 16 billion data points every time we wanted to do this analysis, because that's too much. And then applied the sampling method to both our data set and then another widely used data set, the Q9 data set, to see how well the results would be reproduced and what differences would occur, and it might tell us some interesting things about the chemistry. So without getting too much into the nitty-gritties, what you need to know is this plot here shows all of the measured similarity measures, and this is our developed score. Better scores or lower scores, okay? So the blue line shows the 16 billion data point line for so how well each similarity measure performed. And then the red shows our efficient sampling method. So we do well capture the same thing as the 16 billion data points. And there is some distinction between some scores showing better results than other scores, definitely. This right here shows the comparison of the results for our data set in red and the QM9 data set in blue. And you can see that several of the similarity scores were similar, right? But there is some discrepancies, which I think is very interesting because the QM9 data set is fairly different than our data set. Our data set was pi conjugated electroactive systems. The keyword here is pi conjugation. And the QM9 has a lot of non-conjugated systems. And that lack of conjugation, I think, would probably affect what features are more likely to influence electronic structure properties. So it's interesting that they're both organic. We have some similarity, but there are some discrepancies here. All right, so to this point, I've pretty much just discussed computational data. So databases and high throughput workflows and analyzing computational data. But like I alluded to at the beginning, there is a place for experiment. It is very important. And if we want to use data-driven approaches and experiment, we need large-scale experimental data. And since most of you grad students don't want to like triple your time in grad school, we need another way to generate large-scale experimental data, right? And our answer to that is robotics. So we want to be able to have some idea for a chemical procedure that a chemist might have, a PI may have, and then have a robot just do it, right? The problem is that most chemists, probably myself included, couldn't very easily just have an idea and give the robot the exact instructions for how to perform that experiment. So we have to have some sort of connection. So I developed several pieces of software to help make that connection. The first is Xflow, which is a web-based interface where a user can use drop-down menus and fill in the blanks to create a step-by-step chemical procedure. And it's intuitive for users who have no coding experience whatsoever. Then I created an app on the robot workstation machine right here, where a robot technician can load a procedure and then set several default parameters like where different reagents are and what they are on the table. And then finally, a robotic API that translates the procedure and all of the technician information into actual movement of the robotic arm and measurements from the instruments and whatnot. This entire process also includes automatic data parsers and calculators. So as soon as an experiment is performed by the robot, all the data is processed and extracted, and properties of interest are calculated and presented to the user. So the hours of staring at a spreadsheet trying to figure out how to analyze your data isn't happening here. It all just gets done. So to look a little bit more closely at the Xflow app, or the website, where users can encode a procedure, you might imagine that a chemist has an idea for an experiment, just a very basic cyclical voltammetry experiment here, because that's what we use a lot in electrochemistry. We start with these different reagents, some redox active molecules, some solvent, and our first action is to transfer the solid to a beaker. Second action is to transfer liquid. Third action is to heat and stir it. Fourth action is to do the data collection, right? And on the website, that would look like a sequence of events. Step 1, transfer solid. Step 2, transfer liquid. Step 3, transfer or heat and stir, with the start and end conditions like that. And that goes on farther. I just couldn't fit it on the page. And this is easy for users to create and then also clone to create similar workflows. And then if you want to perform this experiment multiple times, changing maybe one parameter, for example, you may want the robotic workflow to run this 3 different times, and each time you change the amount of solid, you can use this Excel sheet type application I have on the website to quickly do that, and it creates the entire workflow for you that you can download and then use with the robot. So what does this look like in practice? Well, the first thing we did when we built this software and the system was to try to reproduce some literature reported results to make sure that everything was working correctly, that the data parsers were working correctly, the calculators, and so on. So we took 8 well-known redox active systems. They're all organic except for kerosene, which is standard in electrochemistry. And we performed robotic experiments to calculate oxidation potential and diffusion coefficient. And you can see here the comparison of our experiments and the literature reported results, and they both had very good alignment. So we validate that the system is working well. Now, this looks like maybe not so much data, but it's a lot of data. This is actually all the data that went into that plot that I just showed you. So you can see there's about 80 electrochemical measurements here, and it took about 4.5 hours of robotic experimentation. So pretty good speed up on time. And this includes all of the data processing, right? Now we've been expanding the robotic hardware setup dramatically since we did that. Now we have, of course, the robotic arm. We have a vial stand for all of our experiment vials, but we also have 4 different liquid dispensing pumps. We can dispense 4 different solutions into our vials. We have a balance, stir plate, and then a pipette, so we can do density measurements of the solution as well as extracting different solutions. And then we have 3 different measurement stations, one for cyclic voltammetry with macro electrodes, one cyclic voltammetry with ultra micro electrodes, and one for measuring conductivity. This is the robot in action. It is going to multitask. So this experiment just ran. It's going to take that vial away and then rinse the electrodes with the rinsing vial right here. And then meanwhile, it's going to weigh the vial and dispense some liquid. Then weigh it again to figure out the mass of liquid dispense. This will stir it. There's magnetic stir bars in each vial. And then after this, it's going to extract a precise amount of liquid with the pipette, weigh the vial again, so we can know the exact density of the solution we've just created. It'll take away the rinsing vial here, and then put the experiment vial in the conductivity elevator for a conductivity measurement. And this video was actually taken when we only had 2 elevators. We have 3 now, so there are 3 different measurement stations we can do. So how can we do this? Use this to do real chemistry? Well, we are currently working on a study to measure the concentration dependence of several different the concentration dependence of redox properties, given the concentration of redox active molecule as well as the supporting electrolyte in solution. And this is important for those flow battery applications. And to do this, we're using serial dilution of the robot. So we start with a highly concentrated solutions, dilute, do measurements, dilute, do measurements, and so on. And you can see we've been able to do some pretty interesting things and see some good trends in the data. So as we increase the redox concentration, which is always at a 1 to 5 molar ratio with the supporting electrolyte, we see an increase in conductivity before it plateaus off. We can also look at the diffusion coefficient and charge transfer rates for this data. And we can see that as we increase concentration, the diffusion coefficients and charge transfer rates decrease, which may have something to do with the plateauing of the conductivity. And this is replicated with the ultramicroelectrode data right here. And all of this aligns well with similar systems that have been reported in the literature. So that's been really useful. Now, all this data here, this is a lot of data. It took about 6 hours to run all of those experiments. It's about 216 electrochemical measurements with all of the associated data processing. So you can see how we can really rapidly create lots of large-scale experimental data with this. And I mean, just in the past week, we probably created a couple hundred more measurements worth of data from running these experiments. And we just run these experiments on Tuesdays and Thursdays, so things can go very quickly if we wanted them to. All right, so I'll close out with a bit of perspective. Today, I've discussed how we've been able to develop and populate and use chemical data, integrating data-driven design with chemist and chemical intuition. I began by discussing how we organize chemical data, incorporating a philosophical and historical perspective. Then I gave a real-life example of how we've built a chemical database and then used it for various different applications. And I closed with how we can generate large-scale experimental data with robotic experimentations. So ultimately, if we continue to refine and develop these tools, we can build a foundation for automated study and discovery of electroactive high conjugated molecules moving forward. So in the future, of course, we should continue using the data we have. We have a lot of data. So continue our machine learning applications, developing chemical spaces, exploring those spaces, but also considering the philosophical and historical context of those explorations, especially what embedded values may exist in the data structures we have used because we chose function-focused data sets. I think there should also be a very big emphasis on maintenance and availability. I've built a lot of data structures and data analysis structures. We need to make sure they're well documented and are able to be used in the future. And of course, we should continue to develop the robotic platform, do more experiments, and eventually move towards autonomous experiments. This is where not only is the robot automating experimentation, but autonomously deciding what to do next. And this can lead to very rapid production of chemical data and exploration of chemical space. With that, I'll share a list of the publications that exist from this work, as well as those that are submitted and in preparation. And I guess there's a few more that I missed here. And of course, I want to acknowledge everyone who helped make this possible, all of my funding sources from the NSF, several fellowships, and the Center for Applied Energy Research, as well as the Details Project, which funded the majority of this work. And my supervisor, Chad Risco, my committee, the computational support team here at UK have been invaluable, and all the members of the Risco lab and my amazing collaborators would not have been possible to do all of this without all of you. With that, I'll take any questions.
This is the time for the audience to ask questions. We'll have the committee wait until the real grilling begins. So, grad students, yes, questions?
Well, first of all, I absolutely love this. So in your first part, where you talked about the neutral angle for that small library molecule. Well, I was mostly wondering if there was anything you learned about like those specific molecules of that library. Like you mentioned that paper that did like the DFT analysis with the potential energy surfaces, and they talked about things like the end-to-end. But wouldn't the polymers kind of already know which of those guys have the largest end-to-end the manufacturing method to make those polymers? And likewise, could a polymer kind of look at those molecules and tell you which guys have what potential energy surfaces based on like the sulfur? You already know that E3HC does that.
Yeah. Okay, thank you. There's a couple questions in there, so I'm going to try to answer all of them. Thanks for coming, though. Glad you're here. So the first question was there anything else we learned about the chemistry of the systems? And yes, there's actually, that was a third of the paper that I just showed you. The first 2/3 were all about what we learned about the chemistry. So things about what the sterics did and what the electro, whether we had donating or accepting substituents and rings and all that kind of stuff. So there's a whole discussion there. A lot of that is stuff that we mostly already knew, but we were able to demonstrate it in a systematic way. And then you're right, a lot of the classification of these potential energy surfaces, a well-informed chemist could probably do that fairly well. I think the key here is that it's automated. So you don't have to have someone sitting there looking at the structures and making a guess. We could run this on, several thousand, a million potential structures and get the estimation for these mesoscale properties as a scalar value too, not just like which one's bigger, but an actual scalar value. Did that answer all your questions or? Yeah.
Okay, sorry. So in your similarity talk, you showed that some distance functions have largely between 2 and 9 and the high conjugated that set. However, for the people that really care about electronic properties, like material scientists, how well do those distance functions do for subtle differences with high conjugated structures? Like you can have pretty big electronic property value differences for really similar molecules just by breaking the conjugation. And with Judy's fingerprint, that's a really subtle difference.
Yeah, and none of the similarity measures do that well. And actually probably a follow-up paper here would be to develop our own similarity measures that did well. We were trying to just kind of measure how well the existing ones did. And so I think, yeah, that's a good point. And that's something that we should follow up on.
You want to say something about Tanimoto?
Yeah. So, okay, those of you who are computationalists know the Tanimoto similarity measure. Yeah, Morgan fingerprints and Tanimoto. That was actually one of the worst performing for the pi conjugated systems. That is the most commonly used one in literature right now. So, interesting. There's space for us to do these discussions and do these analysis because we're using the bad one. So, yeah.
No.
Yeah.
Really awesome talk. I love it. In my own research, I find I get too excited creating data, not necessarily analyzing the data. And when I see your 90 runs, I kind of get information overloaded. Could you talk a little bit how it was working with all of that? If you ever felt that you were overwhelmed with all the stuff that you had to analyze, or was it you went into the project knowing what you wanted out of it?
So the answer to that is no, because I built the process just before I got the data.
Okay.
So you saw the 90 runs or whatever, but then you also saw on the previous slide, scatter plot, that was able to be created before the data existed.
So
right?
There was never any analysis you did after the data was collected. It was always you went into the experiment knowing exactly what you wanted.
Yes, as far as analysis, as far as gathering data. Now, once we had the data gathered, we looked at it, we tried to interpret it. We oftentimes found things that didn't make sense and then went back and had to redo the hardware because the hardware wasn't working right. So that analysis existed. But as far as compiling the data in certain ways, for the most part, that's all automated at this point. So yeah.
That's awesome. Thank you.
And reproducibility.
And yes, and it's reproducible. We're able to reproduce it well, which is not something that can be said for a lot of things in the literature. Also, to note, the analyses I'm doing here are pretty well established. I'm doing cyclical telemetry and finding like oxidation potential. That's like, you learn that in Gen Chem, right? So I didn't have to come up with some new way to do the analysis, which was helpful. Oh, sorry.
Yeah, how packaged, well packaged was your communication between the robot arm and your program? Did you buy a commercial package that's robot arm connected to?
No, that was all me.
Yeah.
So I think it's packaged well. I don't know. You'll have to ask Zach. He's had to learn it all using my packages, right? But for the most part, besides whenever I have to develop a new, add a new module or develop a new action for the robot, everything can be done with drop-down menus, pretty much. And I built an app on the local machine, so the procedure itself can be assigned fully through my website that I developed, and then it can be loaded and run fully through a drop-down menu app.
I'm talking about the metrics that the robot arm has to actually do to take the vial.
Oh, yes, okay.
Position A, B.
Those.
Bring it up to position A.
So there's a little bit of embedded software in the robot arm itself.
That's what I'm asking.
Yes. So if I have a predefined, like, it's called a snapshot location of the robot, that is predefined for everything on there. And then I tell the robot, okay, move in the Z direction this much to go up, and then move back to this home position, which is a predefined place, and then move forward. So yes, there's definitely some existing software that's in the robot arm itself, but we didn't buy anything external.
What would be really nice is to have some visual.
Yes, so I have a branch of software that is currently working on that.
Oh, that's brilliant.
There is a camera on the robot arm, and I'm working. I can't get the robot to figure out scale and distance, but anyways.
Oh, you need two cameras for binocular vision.
Yes, well, we need to buy that. But we haven't ordered that, right? Also, I'm leaving. Someone else might do that.
But there's work going on with folks in mechanical engineering.
Yeah, so we have some mechanical
That's
engineering
really exciting.
collaborators.
That too.
Yeah.
I think Donna.
Yes.
I wanted to ask you said that the details database accepts like submissions.
Yes.
And I wanted to ask if you've gotten any.
Yes, definitely. Some people in this room, I think, have submitted some things today. Is that not a whole lot?
But like computational or experimental.
Oh, okay. I hear what you're saying now. So when I say user submissions, I'm referring to structures only. So it accepts suggested structures, and then I actually have to go and approve them all. And then they go through my computational workflow. At one point, it actually did accept experimental data and computational data, and it was tagged as something different because I want to make sure I know which theory, but what things were calculated with what theory. But that's actually been taken down because nobody used it. And it made more sense just to have one cohesive data set. Yeah.
I think we really brilliant robotic arms in the city. So how's the standing error? Is there even an error between the two duplicates of the multiple?
How is there an error?
Yeah.
So there are multiple vials with multiple so each point is a different trial with a different vial. So especially for the last set of data I showed you, you'll notice there's a bit of an error in the y-axis for the actual values. There's also an error in the x-axis, and that's because the liquid dispensing pump is not super exact. There's a slight microsecond delay in the communication to stop the pump. So we do have slightly different volumes that are dispensed for each one, and we have an actual person who dispenses the solids, and maybe he doesn't get exactly the same thing every time, and we record that. So the actual concentrations could be slightly off as well. But that's all compensated for because we measure the mass and we do the density measurement to account for volume expansion and things like that. So they're not going to all have the same values. And then also there's just some error in electrochemistry that shows up everywhere. So that's why we do 3 trials. Yeah.
And just to add to part of this last, we found errors from our human collaborators.
Yes.
That's published.
Yes.
Um,
Plural too, actually.
Yeah.
Yeah.
So, you know, there's a whole discussion, right, on reproducibility in science, and you know, there's always questions of how good somebody's hands are at doing something. With the robot, in principle, you should be able to get the same thing in principle.
At least the same error.
The same.
Well, no, actually, we have a really good example of that. So, whenever we do the conductivity measurement, the conductivity probe is a little bit finicky, right? That's what you call in the lab. And the vial elevator goes up, the conductivity probe sits for how long? 20 seconds? We did that and it didn't work well. 30 seconds? No, we have it sit for 60 seconds. And that's what it was. It was the solution had to settle for 60 seconds. The air bubbles had to come out of the conductivity probe. No grad student is going to record, I let the probe sit for 60 seconds in their lab notebook. But the robot, you have to, because you have to tell it exactly what to do. So we're able to quantify that. Yeah.
You mentioned fair data. What does that mean for
Yes.
chemistry data?
So the fair data principles are like a scientific data principles. Those are the data should be findable, should be accessible. Meaning people should be able to get it through the right FSH protocols and stuff. Interoperable, which means you should be able to communicate, or relate what one data in one data set is to another data set, and then reusable. So you should be able to reuse it for multiple things. Yeah. So findable, accessible, interoperable, and reusable.
So what does that mean for your pi conjugated system or
Yeah.
for other chemical?
So the findable part, so my data has a schema, it has a set organization that is well documented. So a number's not just lying in a spreadsheet somewhere. It is a number in a specific organizational format that has some information attached that says this is in electron volts and it represents this value and it was calculated in this way. And you can search through the data set to get there. And it uses standard procedures for calculating certain values and properties, so it can be interoperable with other data sets. It's accessible through the websites and all the different API protocols, and we can reuse it over and over again because I have all of the theory associated with it. So, yeah. It's surprising how much data is not fair.
I think one last question.
Yeah.
So the CV studies you did with the robot and the programming are really awesome, primarily just because they help reduce the possibility for not only user error, but they reduce the amount of uncertainty you can have in the measurements. Like you said, for instance, not letting your probe sit for 60 seconds after you take a CV measurement. There are other analytical avenues, like for instance, UV vis or gas chromatography, where that same issue still applies, especially with concentration, where it's so sensitive that way. Do you have any ideas or suggestions to apply this toward other analytical?
Pretty sure we
positions
have a
such as
UV instrument sitting in the lab.
Yeah.
It's just waiting around for Zach to program it. Yeah, no, the answer to that is yeah, definitely. And we also need the right funding to fund the right instrument to do the right projects, but yeah, for sure.
Everybody knows it when you get funding sources.
Yeah. But also all the software I built, it's very modular. So it's really easy to add an additional instrument, to add an additional action. So when we figured out we needed to be able to measure density of solutions with the pipette, we had to add a whole pipette to the system. CMAP had to build the pipette, but once it was built, it took me half a day to write the code and incorporate it into the system because it's a very modular setup. So yes, it's built to expand.
So I think with that, let's thank Rebekah. What we'll do now is we'll take like 5 minutes as a break. Everybody but the committee has to leave. So please go out and enjoy your day, and then yeah, we'll release Rebekah when she's done.